do this. Okay, afternoon, everyone. My name is Patrick Arvesperis. I'm an undergrad at UC Davis, but I participated on UCSF's iGEM team for 2006. And our team this year, the project for our team was to remotely control bacterial chemotaxis. So just like in our little animation here, what we're trying to do is be able to directly signal or some type of indirect or some type of remote signal actually to be able to control chemotaxis in E. coli. Now, we can't exactly get an E. coli to move left, right, forward, and back like we want it to. But actually, E. coli, what they can do is that they can get their flagella to move counterclockwise or clockwise. And with counterclockwise flagellar motion, you get a glide or a directed swimming, just a little smoother. And if it moves clockwise, it's gonna tumble randomly in all sorts of directions. So you can see in that second, I don't have a laser pointer, but you can see that little gradient there of color. You can imagine that red to be an attractant of some sort. And you can see that E. coli, it's going to glide. It's going to bias its motion to glide more than it tumbles up that concentration gradient. And you can see that in an absence of a gradient, that is on like saturation of your attractant, the far red side of our little picture there, that it's still going to chemotax, but it's going to chemotax in all directions because it's receiving a signal in all directions, and there's no bias there. But you can also see that in the absence of a gradient, in the very bottom picture, it's just going to be tumbling all over the place, and it's not going to be chemotaxing anywhere. So how do we actually observe this chemotaxis? Well, the Goulian lab, they came up with this assay, which is a real nice little assay, where all you have to do is spot cells on a plate and just look at the growth of the colonies. So we tested this out, and what we saw was that in that first picture, there's no attractant and there's no chemotaxis. So our colonies are nice and small because they're not gliding every which way. In the middle picture, what you can see is a saturation of attractant. So they're chemotaxing, but in every single direction. And you can also see that the growth of the colony corresponds to that, where it's out in every single direction. And at the very bottom there, you can see a gradient. On the right side, you can see growth of the colony towards that gradient, indicating chemotaxis. So how does this actually work? Well, chemotaxis is a very complex regulatory network, and it's already been very well characterized time and time again. But pretty much the only parts that we're concerned with are the receptors at the very top there, and our little adapter molecule, KW. Now, when a attractant binds to our receptor, KW is also going to bind, and that's going to act as an adapter, and it's going to allow for all the downstream molecules, downstream targets to go ahead and activate flagellar rotation and chemotaxis. Now you can imagine if you were to remove KW, and then you wouldn't see that chemotaxis anymore because you wouldn't have a way to link up your receptor or get your signal and turn that into an actual movement or a flagellar rotation and therefore chemotaxis. Now, instead of actually knocking out KW, what you can do is that you can mutate KW. And what you would expect in that mutated KW is that that interaction between your receptor and your adapter molecule KW would no longer exist, and you would lose chemotaxis. What you can also do is that you can mutate your receptor, and you won't see chemotaxis anymore because, again, you'll be disrupting an interaction between receptor and adapter molecule. Now, in the literature at the Parkinson lab, what they did was they already identified such an example where they mutated the serine receptor, and that's top right picture, very top bar. And in that mutant receptor with wild type KW, you see a significant loss in chemotaxis. It's reduced to less than a quarter. And then when they mutated KW, but they still had a wild type serine receptor, it reduced it even more. But then this complementary mutation of a mutant serine receptor and a mutant KW, you can see that there is significant restoration of function. And that's also illustrated on the left side, on the far right side there, where we have a restoration of that interaction between the serine receptor and QW. Now, they did this in the serine receptor, and we were looking at the aspartate receptor. So what we did was that we mapped these mutations onto a TAR, which is the aspartate receptor. And then we docked these two proteins up computationally. You can see in the bottom right picture that those mutated residues are still interacting. And these two mutations and their interaction sets up the basis for the orthogonal interaction that we're going to be looking at. And from here, Matt Eames is going to be taking over to tell you more about that.
Thanks, Patrick. So we were trying to test whether or not this orthogonal interaction would work in our particular system where we're using the aspartate receptor. So the first step we did is we tried to throw in some of these constituent components and see how our cells chemotaxed. Here we have just merely QW in our system with no receptors whatsoever. And you see when it's placed in the aspartate gradient, you have no directed motility. Now you throw the aspartate receptor back in there, and sure enough, you have this highly directed growth up the gradient, very asymmetrical, very elliptical in geometry, which is what you would expect in standard chemotaxis. But if we were to put in the mutant aspartate receptor that we had seen and we had mapped onto from the Parkinson's lab serine receptor mutation, what we do in fact see is a vast reduction in chemotaxis, which basically accounts for about a 40% recovery, or 40% of the wild-type chemotaxis. So what we can imagine is there are many receptors, for starters, in the wild-type E. coli, and there's also a bunch of artificially made ones as well through either computational design or directed evolution. All these have unique sensing capabilities, but they all are limited by the same, the same bottleneck of signaling, and that is they all have to go through the same KW protein in order to get their signal downstream. So we can imagine if we redesign this interaction between the signaling domain of the receptor and KW, we can discriminate between many different receptor signals simultaneously. So by redesigning this interaction, we could conceivably come up with signal resolution for as many signals as there are receptors. So with theoretically as many redesigned interfaces as we so choose. So for our case, we'll take the aspartate receptor and the phenylalanine receptor, which is in the wild-type cell, normally a repellent, but in our cases, it's actually been directly evolved, taken from the Goulian lab to respond to phenylalanine. We plug these two into our cells, and of course, we have to also use a codon randomization technique, which will help avoid any unwanted recombination events, given the high sequence similarity between the constituent proteins. So to get this all up and running, we're going to have to strip down our E. coli cell. We're going to knock out all of the original wild-type receptors, all 5 of them. These could normally still modulate the phosphorylation state of some of the downstream signaling molecules like Ki, even in the absence of a gradient. Additionally, we went ahead and knocked out KW, which, since we're going to want to selectively toggle our KW variants to specifically respond to the different signals generated by the 2 different receptors, we're going to want to knock that out of the system as well. And then we're going to want to express these 2 off of a plasmid. So we're ripping this nose off the Sphinx and putting our own funny nose on. So to toggle expression back and forth between the 2 different KWs, we're going to implement an already built switch by the Kiesling and Arkin lab, which is, could theoretically be activated by many different signals, light, radiation, chemical signal. For our purposes, we wanted to just test whether or not this system would work inside our knockout strain. So we put the recognition sequence of the invertase between a red fluorescent molecule and a green fluorescent molecule activated by arabinose, the switching mechanism. For the first part, it just seemed to display exactly what it wanted to, two distinct phenotypes. In the absence of arabinose, we have high red fluorescence. In the addition of arabinose, we have green. But we found that this signal tended to become more stochastic as it went past the early log phase of growth. And we had a number of explanations for this. One is that the plasmid is unstable. The other is that the strain may contain some of the machinery already existing in the switch, like FIM-E or FIM-B. And this might explain some of the characteristic bullseye patterns where we get concentric circles of coloration in some of our colonies. But regardless, we went ahead and built the system anyways with the switch. So all in all, we have two receptors, one for phenylalanine, one for aspartate, and the aspartate has been mutated to interact with our non-native KW. And then we have the switching machinery and then the recognition sequence for the promoter between the two different KWs between those two. So we can toggle expression back and forth of the KWs and decide which receptor we want to be activated. So in our scenario here, in our aspartate negative cell, we see phenylalanine. Our switch is set to KW, wild-type KW, which interacts with the phenylalanine receptor, and we chemotax towards phenylalanine. Conversely, we have the arabinose plus system, which is expressing the non-native KW, which interacts with the aspartate receptor. And in the presence of an aspartate gradient, we're going to chemotax towards aspartate. So how did it all come together in the end? Well, we took these pictures literally hours before we got on the plane yesterday. So it's a little premature, but the results were very interesting. We learned two things. One, we were able to recover chemotaxis with our orthogonal interaction. So in our arabinose plus system, we have the non-native KW interacting with the non-native signaling domain of aspartate, and we are still chemotaxing upwards towards aspartate. And this proves two things. One, that we do recover our chemotaxis, and two, our switch does seem to be working in this system because, as we see, we are chemotaxing towards aspartate, but we're not chemotaxing towards phenylalanine. Unfortunately, in the arabinose negative system, we don't seem to be having any directed motility. And I mean, it's still a little early to tell, but there's two principal hypotheses for why this could be the case. One is the more professionally unsafe method of blaming someone, and that would be saying, well, our phenylalanine receptor that we got from another lab didn't work, which is probably a little bit dubious. But the other safer way of saying it is maybe our KW wasn't successfully cloned into the system. But we feel fairly confident that this is a bug we could work out quite easily in the future because this is a rather a low-level demand of native KW interacting with a native cytoplasmic domain of a receptor. So we should expect that we should be able to recover this chemotactic ability in the future and have a two-state system. So some of our conclusions that we want to throw out there are that these orthogonal pathways do exist that we can incorporate into a chemotaxis system. Ultimately, we'd hope to be able to get many more simultaneous, maybe all 5 native receptors being toggled back and forth between. This is a very preliminary destabilization and recovery interface mutation, but these can obviously be further refined through computational techniques. Furthermore, the switch went ahead and worked in our chassis, which, although was having some setbacks, just looking through the RFP, GFP expression, it still was able to at least toggle towards towards the aspartate receptor. And the chassis, without any of the chemotactic machinery in it, is not going to do anything, but when you put it back into the system, you get it. There are going to be switch performance issues in the future that we're going to try to work out, of course. And the last thing we want to mention is that we could ultimately control this directed motility by other inputs aside from the arabinose input signal. We could do some sort of light activation, which would be kind of a jazzy way of manipulating the system between directional control. So with that, I'll open up the floor. So that's something that could be, what's that? Oh, well, he's asking will we eventually be able to incorporate some sort of switching mechanism which is reversible? Yeah, that's very conceivable. I mean, we're talking about, right, so this was just sort of a proof of principle. Can we get a switching at all, switching activity? I mean, there's a lot more refinements we could make in the future. For example, we opted to toggle between KW expression and not receptor expression because those are membrane proteins and those have slower turnover rates. And if we wanted to get something that could toggle back and forth fairly quickly, KW is a very small protein, which could do that. We could also add on degradation tags and elaborate upon the switching mechanism and get more refined control. But this is just first step in the process. Thanks. Yeah. Oh. So the next obvious steps that I can think of would be to try to first maybe try to put in a safer receptor. We went with the phenylalanine receptor, which we hadn't personally characterized. So something like a native receptor, which we could use the serine or some other system that we know to work. That'd be the simplest way of seeing if we can get the two-stage system up and running. Further adjustments would be the use of computational protocols to elaborate that orthogonal interface, because we're talking about single point mutation destabilization recovery, which is not going to give you the kind of performance that you want ultimately. What they had characterized in the Parkinson lab 15 years ago when they were just trying to prove that these proteins interact. You're talking about 70% recovery, and even with the non-native interactions, you're getting still some noise there, significant noise. So there's a lot of directions we'd like to improve upon this with. Mm-hmm. Sure. Well, it would be, you could, okay, so let's say we could do what we propose to do in the long run, which is create a system that can discriminate between 5 different signals simultaneously, or, well, you could have more complex arithmetic being computed by your multiple different signals. You could require 2 signals to do this and 1 signal from that, or not 1. And so there's further, logic you can add to the system. Then there's conditions that you can conditions you can impose. Right now, it's, since it's in its infancy, we're more interested in a proof of principle, but you could imagine situations where you want your bacteria to swim towards something, and when they get to that something, they go somewhere else. So you could have, if your system is chemotaxing towards aspartate, you could have aspartate being the mechanism that causes the switching event. And so when it finally gets there to destination 1, it can now, you could have it run the base